Hello, distinguished guests. I am Dr. Deepa Bhartiya from Tzar Labs, Mumbai. At the very outset, I would like to thank the organizers for this opportunity to share our work with all of you. Before joining Tzar Labs, I spent almost 2 decades working and heading the Department of Stem Cell Biology at National Institute for Research in Reproductive Health, which is a premier institute of Indian Council of Medical Research based in Mumbai. Here, we were working on tissue resident stem cells, which play a crucial role in maintaining lifelong tissue homeostasis and how their dysfunctions contribute to various pathologies, including cancer. Building on this foundation, now at Tzar Labs, we are into early detection of cancer and we study these stem cells in circulation, besides the existing focus on circulating tumor cells and ctDNA. And obviously, the studying stem cells has much more advantage compared to that I'll be sharing in my ahead presentation. If you see what is cancer, cancer is a prehistoric disease. Even dinosaurs had cancer. But it is really saddening to realize that despite being a prehistoric disease, still today we do not understand how cancer initiates and why it recurs despite so many advances in cancer treatments. Why patients with even immunotherapy show therapeutic resistance. So, almost 100 years ago, cancer was being treated by surgery, by radiotherapy, by chemotherapy. But these were always associated with recurrence. And it was thought that one size shoe does not fit all, and therefore precision medicine was introduced. Patient specific mutations were targeted. But this also did not help because what happens is when you target a particular somatic mutation, another clone appears which is more malignant. Why is this happening? We do not understand yet. And as a result, eventually the immunotherapy was introduced with lot of hope that maybe this is a possible cure for cancer. But now we have realized that even immunotherapy, CAR T also, is associated with recurrence. So that is why there are several unanswered questions in the field of cancer biology, which I have listed here. Why there is no cure for cancer as yet, even though we are treating it for almost more than 100 years by now? Why precision and personalized medicine has not been very helpful? Why do oncologists still struggle to treat and overcome recurrence relapse? Why targeting somatic mutations does not help cancer patients? Why newer and more aggressive clones appear all the time? Why is there therapeutic resistance in response to immunotherapy? So, the problem actually, if we look at it overall, is that there is still no consensus on how cancer initiates. Cancer was treated as a genetic disease for almost 100 years. But now, in recent times, this concept is being challenged, and people understand that genetic mutations may be a consequence and not a cause of cancer. The biggest dilemma is what cells survive cancer therapy and cause recurrence. Because it is not the primary tumor, but the one that is occurring because of metastasis and recurrence that is responsible more than 90% of deaths. Can we target these residual cells? And it is really very frustrating that despite deep molecular remission, MRD negative leukemic patients also show relapse. Studying relapse and residual disease in solid cancers is a big, huge challenge. Mostly it is nowadays analyzed by looking at CTCs and ctDNA. But CTCs, they are sloughed off cells from the surface of the tumor. How can they inform about the residual disease in the particular patient? This is CTC, ctDNA is a non invasive approach to study residual disease, besides studying somatic mutations or by doing immunophenotyping of aberrant cells in the tissue biopsy, which is only possible once. So, against this backdrop, what I would like to discuss today are novel tissue resident pluripotent stem cells termed very small embryonic like stem cells VSELs, which play a crucial role in maintaining tissue homeostasis, and it is their dysfunction that results in various pathologies, including cancer. Now, if you here, we'll discuss these VSCs briefly. Professor Radzak from University of Louisville was the first to report these stem cells. They are the most primitive cells that sit at the top of cellular hierarchy in all tissues. They exist in very small numbers, virtually immortal, and they survive throughout life. They play a crucial role in regular turnover of cells, ensure endogenous regeneration upon chronic injury, and VSEL dysfunctions initiate all types of hematologic and solid cancers. Here is a picture. The VSEL is a small cell. It's a very small stem cell compared to hematopoietic stem cells, which are very well studied by globally. Now, if you look and compare the two cell types, the VSEL has lot of euchromatin. It is small, high nucleocytoplasmic ratio. There is minimal cytoplasm and very few cytoplasmic organelles. And almost 80 independent groups globally have confirmed their presence. VSCs, as shown in this particular slide, they exist in multiple organs and are developmentally linked to primordial germ cells. Now, what we all know very well that during early development, the primordial germ cells migrate from the dorsal ridge to the genital ridges, where they survive throughout life as they differentiate into germ cells. But what we believe now that these stem cells do not go only to the gonadal ridges, but they mobilize to all the organs in the body and they survive in small numbers throughout all the tissues. As shown in this pie chart also, in different organs in the mouse organs, you can see these stem cells are present and they are present in variable numbers. And it is surprising that the brain has large number of VSCLs. And this reference at the bottom show is a recent review on basic concepts, whatever is known about VSCLs. Now, in order to see VSCLs, it is you need to see and believe in what you see. So, if you look at a normal mouse seminiferous tubule, here I am showing you the immunostaining for PCNA. And normally, what we find that there are germ cells at the bottom, Sertoli cells, and then there is spermatogenesis towards the lumen. But if you treat the mouse with FSH, and within 12 hours, if you study, these small stem cells, they lit up and they cannot be missed. You see them very distinctly. They are positive for PCNA, they are dividing, and they are very much there throughout the seminiferous tubule. These cells, they are expressing nuclear OCT4A, as you see in the right panel, on the top right panel, whereas the spermatogonial progenitors at the bottom are having cytoplasmic OCT4. So it is the nuclear expression of OCT4 which marks the VSCLs, whereas the progenitors are marked by cytoplasmic OCT4. These cells are very well studied in the cord blood also. Same way they express CD133, they are CD45 negative, and they express SSCA4, OCT4A. And although the cytoplasm is minimal, but cytoplasm is definitely there, and this is confirmed by the presence of actin expression. Now, these cells are quiescent in nature. They exist in G0 stage of the cell cycle and they are there in the G0. And if you compare with embryonic stem cells in the middle panel, bottom row, embryonic stem cells are in active cell cycle. They are present in all the different stages of cell cycle, whereas CD133 positive and SSCA4 positive cells are only restricted to G0 stage of the cell cycle. Now, if you treat the mice with chemotherapy, these cells are not affected by chemotherapy, which is a very important point that these cells remain unaffected by chemotherapy because of their quiescent nature, and they express various pluripotent markers and also show telomerase activity. One of the most characteristic feature of VSCLs is that they undergo asymmetrical cell divisions. By this, we mean that the stem cell, when it divides into two, the two cells are of different size and different fate. The small cell is for self renewal, whereas the bigger cell is the progenitor, which is having cytoplasmic OCT4 that undergoes symmetrical divisions and clonal expansion before it initiates differentiation to maintain tissue homeostasis. So these are the different cell divisions that are involved in maintaining tissue homeostasis. And on the right side, bottom side, I show all these different type of divisions in mouse endometrium. The reviewer, when he was reviewing this paper, wanted a high magnification image. So you see that I've shown at 100x magnification very clearly that clonal expansion is basically rapid expansion of cells with incomplete cytokinesis. These are not simply aggregates of cells, but these are having cytoplasmic connectivity. And the same thing which I'm showing in the middle panel, the cell divisions, they are also seen in endometrial sections also. We have reported this. Asymmetrical cell division, symmetrical cell division, and clonal expansion. Now, these cells also have they differentiate into epithelial cells. This is a very important function of VSELs. What my student did was that she isolated SSCA1 positive VSELs from mouse uterus of a GFP mouse, a mouse which was constitutively expressing GFP. So the cells she isolated were GFP positive, and when we transplanted them into a wild type mouse uterus, within a same estrus cycle, if we study, we showed that these green VSCs differentiated into epithelial cells, which were also expressing GFP. The specificity of this experiment was based on two negative controls. In one, we used SSCA1 negative cells. So these cells were sitting in the tissue. They did nothing. They did not differentiate into epithelial cells. And of course, the other negative was where primary antibody was omitted and we did not see any staining. So whatever results that I'm showing you are very, very specific, and we are trying to show that it is the VSCLs that differentiate into epithelial cells in all solid tissues. These cells also participate in endogenous regeneration upon chronic injury. We showed this by inflicting mechanical injury into a mouse uterine horn. You know, with a needle, we mobilize inflicted injury into the uterine horn with a needle, and within 24 hours, you see this red circle. A large number of small size VSCs became activated, and we could see them in large numbers. And within 72 hours, these cells differentiate into progenitors, and eventually the endometrium will regenerate. So it is the VSCs which participated in regeneration, but within the first 24 hours, and then they disappear. This bar diagram is also very interesting data. OCT4 is present in high expression at even 24 hours, it reduces by 72 hours. But interestingly, OCT4A, which is specific to VSCLs, is increased at 24 hours and then it becomes normal by 72 hours, meaning that VSCLs are activated upon injury, and once they have done their function, they go back to quiescence. And SCA1 is another stem cell marker which remains elevated during this process of endogenous regeneration. This is another example where a work was done at Tata Hospital, and they showed that when young children with different types of cancers were treated by chemotherapy, radiotherapy, and even surgery, they become totally infertile and azoospermic when they are old. So now we took testicular biopsies of men who were young individual survivors of cancer. And if you see on the right side panel, the testicular tubules are totally devoid of sperm. It is total azoospermia. But if you take these biopsies and make smears out of it, it is really surprising to see that besides the Sertoli cells, there are these stem cells, dark blue spherical cells sitting in that testicular biopsy. So for almost 20 years, these cells were sitting in that testicular biopsy in a quiescent nature, in a quiescent state, and they are definitely there. They express OCT4, cell surface SSCA4. So, what is cancer? What is the genesis of cancer? For us, we think that under normal homeostatic conditions, there is a proper balance between proliferation and differentiation. And cancer is a stem cell disease. So, stem cell dysfunctions result in cancer. There is a disbalance between proliferation and differentiation. It is the epigenetic changes in VSCLs that transform themselves into cancer stem cells. There is a lot of hypomethylation, genomic instability, etc., which is associated with cancer onset. So, what is the most important thing I like to point out is that mutations are a consequence of cancer. They do not initiate cancer, they occur as a consequence of cancer. And we have put forth a theory, tissue resident embryonic stem cell theory for cancer initiation. So, this particular diagram describes what I am trying to say. Under normal conditions, VSCLs are there with abundant eochromatin, and these cells undergo asymmetrical cell divisions to give rise to a bigger progenitor. It is the progenitor cells that undergo symmetrical cell divisions and clonal expansion, followed by differentiation, and this is how tissue homeostasis is maintained. However, in case of cancer, there are epigenetic insults, lots of extrinsic or intrinsic insults which causes epigenetic changes in VSELs. As a result, they come out of their quiescence, enter the cell cycle, and start dividing rapidly. Because of epigenetic changes, the VSELs get transformed into cancer stem cells, and when they undergo rapid clonal expansion, etc., because of genomic instability, they tend to develop mutations. The normal differentiation of these progenitors is affected, so it is block differentiation and excessive proliferation which gets associated with somatic mutations. So this slide shows how VSCs drive cancer. Under normal conditions, it is the VSCs which maintain homeostasis in the epithelial cells. They are responsible for regular turnover of epithelial cells. So there is no need to think of epithelial cells, their de-differentiation or epithelial mesenchymal transition to have a role in cancer. It is these VSCs which are actually getting epigenetically altered, which initiate cancer, which get mobilized, and they will get mobilized to distant regions where they cause metastasis and recurrence. So mostly people think of this middle panel about that cancer is a clonal disease. It is the clonally expanded cells that initiate cancer, but actually a lot more is going on behind, or much more before the clone are forming. It is the VSCs which are actually having a role in cancer initiation. So, what is happening as of today? How do we patients are being treated for cancer? Basically, what is happening that we are trying to treat the bulk cells in a tumor. It is the cells, blue cells that are being treated, but the stem cell is getting spared because of its quiescent nature, etc. Why is it getting protected from immunotherapy? Because they are these cells being pluripotent, they are immune privileged. So they are not even affected by immunotherapy. And when we treat all the large number of cells, but the stem cell survives, it causes recurrence. So there is no need to target multiple other cancer cells. We just need to target the cancer stem cell. So what we are trying to say that cancer is actually a epigenetic disease and it can be reversed. Because we are targeting the wrong target cells, the cancer cells, and not the cancer stem cells, that is the reason why there is no cure of cancer as yet. So, how can we reverse cancer? That is a question which everybody will have in mind. This is possible by use of epidrugs. We can reverse them back to the quiescent state. And also, we need to take care of tumor microenvironment, the niche, where mesenchymal stromal cells, their transplantation, etc., will have a crucial role. So this is the current status of cancer. How people are looking at tumor. They feel that there is a tumor. The epithelial cells are their surface cells. They get sloughed off as cancer CTCs, and these cells die and they release ctDNA. And we can study ctDNA for MRD for early detection. But I am sure that this is not going to give the answers that we are looking for. Actually, tumor is much more than CTCs and ctDNA. We need to look at the stem cells, the epigenetically altered stem cells, which are the cancer stem cells, which develop mutations and they are also present in a tumor. They get mobilized, they are also there in circulation. And that is where our company Tzar is really focusing on. We study these stem cells in circulation and we are trying to give early detection of cancer. These are the best targets to understand residual disease and cause for recurrence, etc. So, in summary, I would like to conclude that VSELs initiate cancer, are responsible for its progress, metastasis, and recurrence. They are more relevant, and their presence challenges the concept of epithelial mesenchymal transition and the concept of dedifferentiation of epithelial cells. I would like to say that it is not the epithelial cells that initiate solid cancers, but it is the stem cells which are located amongst the epithelial cells which are actually responsible for cancer onset. VSELs survive oncotherapy. They survive immunotherapy as they are immune privileged. It is possible to reverse cancer if detected early. Thank you so much. And this shows all the list of all our publications. I'm open for all questions. Thank you.